So, this is our eye looking straight on, okay? These lines, this outside portion here is called the iris of your eye. The pink cells are smooth muscle cells. Then we have proteins. These proteins are encoded in our DNA, and they're located in the iris. So this person happens to have these blue proteins in their iris. This person is going to have blue eyes. That's right. For a person with brown eyes, the only difference is their genes encode for a slightly different protein. So let's say we add these yellow amino acids. So we add these yellow amino acids to our eyes now. This person may have brown eyes, or green eyes, which is a little bit more rare, or hazel eyes, right? Different proteins will have different pigments, and those pigments reflect different colors, make us see different eyes. Eye colors, sorry, not different eyes, different eye colors in the iris. So now the big space in the middle of your iris, what is that called? Does anyone know the black hole? Ben? Pupil, right, that is your pupil. That's right, so the purpose of your iris is when you're in a really bright room, right? So right now lights are on or it's bright outside, that muscle is going to shrink your pupil. So that muscle is going to shrink your pupil. That's going to allow less light into your eye, because at night the opposite happens. So at night, or when it's dark in a room, the smooth muscle in your pupil is going to open up, make your pupil even larger. That's called dilating. Your pupil will dilate, which means we're opening it up. And when we open up, that allows more light into our eyes. So now we're going to switch pictures. Now we're going to this section of the eye. So now this is like an eye cut down the middle, right? And we're looking at half of it, right? So we're looking at half of an eye here. All right? Bam. So first thing I want to do, I want to show you where these pieces are. Iris, these smooth muscles right here. They can close. Can you see that, Chris? They can close, right, or open as we need them, right? So that's just another way of looking at the iris here, right? So they can close. This is constricting, and this is dilating the pupil, right? So now the pupil is right here in the middle. All right, the pupil is right here in the middle. Yeah. So epithelium cells are cells that line your organs, right? So your skin has epithelium in it. Your intestines have epithelium cells. Your blood vessels have epithelium. They're cells that line the outsides of your organs. So your eye also has epithelium cells, all right, around the outside. The epithelium on the cornea is super thin. So thin, in fact, that it looks to us to be totally clear. And clear membrane is this portion here. So it's the epithelium, then there's a little bit of space inside, and then a second layer of epithelium, and it's totally clear to allow light to flow into your eye through your pupil. Also, there's very few blood vessels in your cornea, right? And the reason for that is it gets its nutrients from the fluid that's inside just inside the eye. Because if we got our nutrients from blood vessels in our cornea, they would cloud our vision. Next thing we're going to be talking about another set of smooth muscle, ciliary muscle. This is also a ring that goes around the lens of our eye. So our eye has a lens that allows us to adjust for focusing, for distant objects, for close-up objects, right? we can manipulate the lens. And we manipulate it via this ciliary muscle. So the lens, you need to put it in your notes, is surrounded by an elastic membrane, all right? It's almost like a, from what we heard, is like a pocket. It's almost surrounded by a pocket of this elastic material. When your eye is at rest, that lens is going to be round and globular, all right? When we need to see things that are farther away, we have to change the shape of this lens. So these ciliary muscles will contract. And when they contract, the lens will change shape. Bam. That lens will change shape and it will become more of an oval, more oval shape than it is a round shape. So then when we need to look at something close up, these muscles relax again and the elasticity bulges that lens back out into a circular shape. Inside the lens, what gives it its properties, they are actually protein fibers that are inside the lens. And it's actually, they're not the same fibers, but the acrylic blocks that we used in the lab earlier, was it this week or last week, wherever we did it, are actually also made up of fibers, correct? That's what we found? The acrylic is made up of fibers. So it's the same kind of idea as the acrylic, right? They're different fibers, but they're both a type of fiber in the lens and in the acrylic that's going to change the refraction of light as it goes through. So that's how our lens works. And depending on, we know, different size lenses will have different focal points, will have different properties for refraction. Right? So when we change the shape of that lens, it's going to change how we refract the light. And we'll take a look at that as well. Okay, next thing we need to talk about, the retina is the back of the eye. All right, this is where it's basically the movie screen of our eyeball. All right, in order to see things clearly, we have to project that image clearly on the retina. If the image being produced on the retina, on the screen, is blurry, like when you were moving your screens today, right? If your screen was too close or too far, you saw a blurry image, correct? It's the same idea with your retina. If our eye focuses that light too close or too far away, and you get a blurry image, you're going to see a blurry image. So this is the movie screen of your eyeball. You need to have a clear image on your retina in order to see a clear image. The white cells here, they are called rod cells. They are responsible for identifying lightness and darkness. So white and black, shades of gray, that's what the rod cells will see. If you only had only rod cells, you will see images in black and white only, just light and dark, yes. The red, blue, green are called your cone cells. Your cone cells are responsible for seeing color. Bridget, I'll look that up, sorry. Your optic nerve is the nerve that goes, it takes the signals that your rods and your cone cells are sending, and it sends it to your brain, and that's what sees the picture. Red, green, and blue cone cells, right? And those cone cells, depending on how they're stimulated, we'll perceive every color that we can see, right? Using those three cone cells. So these again are like a circle all the way around your lens. Yeah.
And like the little tube in front of the lens.
These two?
Those are like ciliary muscles too.
These are not ciliary muscles. These are smooth muscles that make up the iris.
Oh, okay.
Right, and it makes so that these contract and relax, right? Which makes what smaller? Not the iris. Well, I guess the iris would be, but yeah, the pupil. Yeah. So the pupil gets smaller and larger depending on how much light is available to your eye. Right. You don't want to have too much light getting to your retinas. Right. But if that light is too intense, then it's going to start you know, damaging the back of your eyes, damaging your retina. So we don't want that. So this mechanism will close when light is really intense, and will open when light is at low levels. So therefore, right, exactly. So depending on where your object is that you want to focus, your ciliary muscles can contract, right, and this will bring the lens with it, and it changes the shape of that lens. We'll either make it more narrow or when it relaxes. So when it's contracted, it makes the lens narrow. When these muscles relax, the lens relaxes and becomes more round, more globular. What it is, yeah. So we have our eye lens, also has a principal axis, also has the median running through it, right? And we have an object. Light rays are coming from that object. They're coming parallel. Pardon me?
Is it supposed to be the same size?
No.
So it gets smaller?
Yeah, it'll get smaller. Remember converging lens, when you have an object that's beyond 2f prime, what's the size of the image going to be?
Yeah.
Yep. It's smaller and it's inverted. When we look at an object that's close up, that's right in front of us, what happens is those rays are going to be coming at us, at our eyes, at an angle that's diverging. All right, it's going to be getting bigger. Because it's up close, those light rays, we see all of the light rays that are spreading apart from that image. Okay? So what happens is when we're looking for something up close, the light rays are diverging, our ciliary muscles relax, and the lens becomes as circular as possible. This increases its refracting power. All right? And it does so accordingly, so that way it can adjust where the image is located on our retina, right? So it wants to put the image right on the back of the retina. And again, so when we have parallel rays coming in, our ciliary muscles contract, making the lens more narrow, right? This decreases its refractive power. So now the light rays don't bend as much, right? And depending on how much our lens refracts, right, that light will put this image at a different location. So our lens changes depending on the angle of light rays hitting our eyes, and the way that those lenses change, right, they change in order to put that image directly on the back of the retina. Farsighted is hyperopia. So remember when I said when we see things closer, the light rays hitting the eye diverge, right? The light rays are coming in at a greater angle. So their ciliary muscles relax, their lens goes to become more globular, but it can't get round enough. It cannot increase its refractive power enough to refract these diverging rays coming in. So what happens is, it refracts it as much as it can, and the image ends up being behind the retina. So we fix it using lenses. So this lens is going to represent glasses. All right, so these are glasses. It's going to converge them. Yeah, exactly. It's going to converge them. So these light rays coming in at an angle are going to hit the lens, and they're going to be converged. And we do this so that they can come in like they're parallel. So we basically trick. It's not a trick. We basically allow our eyes, because we can't get enough refracting power when light rays are diverging, we converge them enough right, from our glasses, that now our lens can get to the shape it needs to. And this is going to be
pretty droopy, right?
and now the image is going to be back on our retina. All right, this works exactly like it should. The problem with nearsightedness, when you get objects that are far away, so objects that are far away, remember, the light rays hit your eyes at parallel. Okay? This refraction, the ciliary muscles contract. It tries its hardest to shape the lens and make the lens as non-circular, as oval as possible, but it can't get there. It can't get there, like the oval part, it doesn't get oval enough. So what happens is,
Yeah, can you make sure that's all in?
Wait.
yes, it still has too much refractive power. So when that happens, you're going to get the image is located in front of the retina. So it converges the light too strongly, and it converges in front of the retina. So again, remember, it gets blurry again, right? So again, so this image is focused in front of the retina. It's blurry. We throw our diverging lens in front. Oh, it's this way. So parallel rays hit the diverging lens, and they diverge out, making it seem like the object is up close, right? So it simulates the light rays coming from an up close object. Now, the ciliary muscles can relax, the lens can become more globular, increase its refracting power, and it's going to be able, again, let's put that image on the back of the retina.